Hey everybody, I'm sorry I can't be with you today, but I'm at my daughter's competition. I'll be with you next class. Today's stuff is not necessarily easy, but it's not overly hard too. So please don't make it harder than what it is. If you have questions along the way, jot them down. We'll go over any questions you have next class. Okay? So I have two beakers here, and they have a coffee filter in each beaker. Okay? And I'm going to go ahead and pour water in the first one. And I'm going to go ahead and pour water in the second one. And I want you to make some observations here. One observation that you might be able to see is that all of the water in this beaker, or in this filter, is able to go through that beaker. Okay, it is called permeable. Permeable means that all substances that are in this membrane is able to go through. Okay, permeable. So I'm going to show you again so you can see. Here's my cell. Here's my cell membrane. This filter is going to act as a cell membrane. Okay, and I'm going to pour a substance, just water, through my cell membrane, and it is going to allow all of that water to go through. I want you to think about when we became the cell and we sat inside and we covered ourselves up in that curtain, and we talked about how membranes let things in and out, that they're not all solid, that there's spaces in between that membrane. And I want you to think about that and think about how water is able to go through that membrane. And because all of that substance goes through, it is called permeable. Now, if I have the same cell, but I have different substances, and I introduce water to it again, you're going to notice that only the water is able to go through. That membrane is semi-permeable. Semi-permeable. That means it only lets a certain substances through and certain substances that don't go through. So in this case, it let water through, but it did not let the larger molecules through, or in this case, the chips. So the difference between permeable, which lets all substances through the membrane, and semi-permeable, which lets some substances through the membrane. So I'm going to go ahead and put this off to the side, and what I want you to do is grab a blue, grab scissors and glue in your spiral notebooks. I'll give everybody about literally 15 seconds. So back in 2 seconds. Alright, there are quite a bit of notes today, so just grin and bear it. Hopefully it's going to only take me about 20 or well, about 30 minutes of notes today, and then you guys have a follow-up lab, okay? So let me get some dry paper, and on the top we're going to go ahead and put the title. I'm going to try to make this a little bit closer so you guys can see it better. There we go. We're going to call this Diffusion and Osmosis. Alright, and we're going to talk about those different parts of it. Okay, before we get started, let's go over some basic vocabulary. So we just talked about, and Mrs. Todd, let Mrs. Todd know when you need her to pause it so you guys can get caught up, okay? So let's talk about permeable. P-E-R-M-E-A-B-L-E, permeable. Permeable means that it allows all substances to go through a membrane. Okay, it can be gases or liquids, and sometimes even small solids. So it just depends what that cell needs and how it's going to get through that membrane. Okay. Then you have semi-permeable. And semi-permeable allows only certain substances to go through a membrane. Okay, and an example of this, so let's do permeable, would be a coffee filter and water. Meaning that only the water is going to go through. Semi-permeable, I want you to think of sand and water, where only the water goes through. Meaning the sand is going to be left behind and not given access to that cell. Okay? Alright, so hopefully everybody is all good with that. Now again, the notes are going to be a little bit long today, so I just need you to grin and bear it, and then after we're done, you guys have a lab to do. Okay? And just make sure that you clean up your lab. Okay, so everybody good? If everybody's good, give Mrs. Todd a thumbs up. That lets her know that you guys are ready to move on. Everybody good? Okay, so let's go ahead and put a little star here. Let me get this up a little bit. There we go. A little star. Just like a factory, items must be brought just like a factory, items must be brought in and out. Okay, so just like a factory, you get mail in, you get packages in, you send packages out. So you have to allow things to come in and out of that factory. Same thing with the cell. Same with cells. Cells have the ability to move particles and water. Whoops, there we go. As it needs them. Okay, now you got to kind of think about this as, if a cell needs water, it's going to bring in water. But when it's bringing in water, it's trying to maintain a balance. Can anybody remember what that word is that tells you that you're trying to maintain an internal state or a balance in that cell? Hands up, please. Starts with an H if you haven't guessed it. Or know it. Shouldn't be guessing, should know. Good. Homeostasis. Homeostasis is the tendency to be equal or to be balanced. Oops, spell balanced right, sorry. Okay, and that was the review from last unit. So remember, when the cell is letting things in and out, it's trying to maintain the balance. If it's high in carbon dioxide, it's going to let that gas out. If it needs oxygen, it's going to bring that in. If it's got too much water, it's going to send some out. And it's going to go in and out through that membrane. And sometimes it's permeable, meaning it's going to let anything in and out. And sometimes it's semi-permeable, where it's only going to let certain things in and not let certain things out. Okay? All right. So let's talk about let's break this down. There are two types of transporting things in and out of the cell. There's two types, so we're going to go through both types. Type number 1 is called passive transport. I know, here's where all the big vocabulary comes in. Passive transport. Please underline it because it's really important. Passive transport. Passive transport is the movement through a membrane or no membrane using no energy. Okay? It is movement through a membrane or no membrane using no energy. So that cell does not need energy for this. Your cell does not need movement for this. Okay? And there are two types of passive transport. I told you everything's going to start to build now. One type of passive transport. Remember, passive transport, no energy. No energy is required. It is real important that that is your key word. Passive transport requires no energy. Okay? Here we go. Sorry, I'm doing the best I can here, guys. Okay, two types of passive transport. The first type is diffusion. Okay, your lab today is going to be on diffusion. Okay, diffusion. Here's an example of diffusion. If you guys had your heads down and not looking, and I cut an orange and squeezed it and kind of took a fork and dug into the rind of the orange, what would you smell at your seats? Yeah, the answer is you're going to smell the orange. That is diffusion. Here's another one. I go and get a pitcher out of my kitchen, fill it with water, and I take a thing of Kool-Aid and I pour the Kool-Aid in, and I watch it start to move in the water and mix so it's equal throughout. That's diffusion. Okay, here's something gross. If you're sitting there and your neighbor passes gas and you smell it, that is diffusion. There is no energy required for those molecules to move around. Okay? So diffusion is the movement of particles from high concentration to low concentration. Okay, it may have a membrane. Okay, and we're going to go through two examples of this. All right, is everybody good with me? Yes? I hope so. All right, so I'm going to go ahead and draw an example here. One example is I'm going to draw a little beaker, and I'm going to draw another beaker right beside it, and then I'm going to draw a beaker right beside it again. Okay, everybody good? All right, and this is with no membrane. Okay, now what I want you to do is I want you to get your blue crayon, or if you have green, that's handy, you can get that. And I'm going to draw with my pencil a water line on each one of the beakers. And I'm going to pretend that I am putting drops of food coloring in. In the beginning, the food coloring is going to be a big clump as you're adding it in, correct? As it's given some time, that food coloring is going to start to spread out. And eventually, that food coloring is going to spread out so that it covers or fills all the water inside there. Okay? That is diffusion. High concentration I have a high concentration of food coloring and a low concentration here. Here, that concentration is starting to spread around a little bit. Those molecules are moving around in here. In here, you're going to notice that it has reached equilibrium. That means it's reached homeostasis, that there's no area of high and low anymore. It's all spread out. Okay? So let me go through what this is again. Passive transport means that there's no energy used from the cell. Diffusion means you're moving particles from a high concentration to a low concentration, in which case high concentration molecules are going to start to spread out. Okay? So here's an example of diffusion with a membrane. So let's say I have a balloon and it's filled with helium. Okay? Not one of those foil balloons, just a regular helium balloon. Now, what happens to a helium balloon over several days? You bring it home, it's all puffy, it's standing straight up, it's a regular great helium balloon, and then, in a couple days, it starts to shrink and starts to lower itself to, at the end, you're going to find a balloon on the ground. Okay? So let's talk about what's happening here. You have high concentration of helium and low concentration on the outside. High on the inside, low on the outside. Now remember, things like to be equal. So over time, that balloon acts as a membrane. And some of the helium is allowed out. Helium leaves. And as that continues, less and less helium is going to be in that balloon because it's wanting to get out. So this is with a membrane. Okay? So you have two examples here. You have one with no membrane, and think of it just adding food coloring into a beaker of water and watch it go ahead and move and spread apart. Then you have a high concentration of helium in a balloon, but over time, that membrane allows the helium to start to leave. And eventually, that helium balloon becomes shriveled up and pretty much is garbage. Okay? Now remember, is this semi-permeable or permeable? It's a great question. Is it letting things in or is it letting things out? So I would say in this case this is semi-permeable. It's letting things out of that membrane, but not necessarily in. Okay? Is everybody good with that? Everybody okay? All right. I'm going to go ahead and flip over my page. All right. So let's talk about the number 2. Again, this is passive transport. Remember, passive transport means that it does not need energy to do this. It just happens because substances go from high to low. All right, number 2. So the first one was diffusion. The second one is called osmosis. Okay? Osmosis is a type of diffusion. With water. It is no other substances besides water that's able to enter that cell or leave that cell. Okay? Only water through a semi-permeable membrane. Okay, a type of diffusion with water through a semi-permeable membrane. Okay, that means only certain items are allowed through. Okay, it's just another definition of semi-permeable. Only certain items are able to go through. And again, it moves from high to low concentration. Okay, and again, no energy is used by the cell to make this happen. All right, it's just part of homeostasis where things like to be equal. All right, so if you have something of high concentration and something of low concentration, high concentration is going to move into the low concentration to balance each other out. Okay, so here's an example of osmosis. So let's say I have a red blood cell and it kind of looks like a frisbee. Okay, and let's say I take this red blood cell and I put it in distilled water. Now, what happens to this red blood cell? This red blood cell will take in water. It will take in water. That's why it is osmosis, because it takes in water through a membrane. It's taking water through this cellular membrane that's on the outside. Okay? Now, what eventually will happen to that cell? Great question. It's eventually going to take in so much water that it's going to get really, really big. And in fact, it can get big enough that it can burst. Okay? So let's go back and go over everything real quick. So I'm going to flip this back over to page 1 of my notes. Permeable allows substance to go through a membrane. Lets any substance go through. It's not picky. Anything can come and go. Semi-permeable only lets certain substances in. Only certain ones. Okay? Passive transport. No energy is required by the cell for this. Instead, it lets things go from high concentration to low concentration. No energy. 2 types of passive transport. One is diffusion. Is the movement of particles from high to low, and may have a membrane. High to low. Think of adding food color to a beaker. Think of a helium balloon sitting in your bedroom. Over a few days, it's going to start to wilt down. And that's because the helium is leaving your balloon and heading out. Because this acts as a semi-permeable membrane. Allows things in and out, or allows things out. Okay? Everybody good? We'll talk about this more, so don't panic if we're lost a little bit. All right, let's flip it over. We're still talking about passive transport. Number 2 is osmosis. Osmosis is a type of diffusion. Don't get this confused. It is a type of diffusion, but it only uses the flow of water through a semi-permeable membrane. Okay? No energy is required. And we'll talk more about this when we review next class. Thumbs up if everybody's ready to go. Thumbs up if everybody's ready to go. Everybody good? Okay. A second example of osmosis would be a plant. So let's say I have a plant. Okay, and there's the ground, and the plant's got some roots. Real quick drawing, nothing super fancy. You got a plant, okay, and the plant doesn't get enough water, so it starts to wilt. Poor little plant starts to wilt. Okay, now I have high concentration water here, and I have low concentration of water here. So what do you think is going to happen? Water is going to move from high to low. That's what brings in the water from the ground to the roots of that plant. Goes from high concentration of water to low concentration in the plant. And eventually, depending on the plant, it will very soon and quickly gather the water it needs to be able to stand itself up again. Remember, inside that cell, in each of those plant cells, is a big vacuole full of water. That water that goes here goes into each cell and fills up that vacuole, stands that plant right back up where it's supposed to be. High to low, going through membranes in the plant, and I'm dealing with water. That's the difference between diffusion and osmosis. Diffusion is moving from high to low. Osmosis is moving from high to low with water. All right. Thumbs up if everybody's good to go, because we're almost done notes already. Told you it would be about half an hour, and I'm pretty close. Okay, so those are the two active, or the two passive transports. Remember, passive transports requires absolutely no energy for that to happen. It's basically moving things from high to low to meet in a middle ground. So let's talk about active transport. Okay, active transport is the movement of substances across a membrane from low concentrations, spell this right, to high concentrations. It's opposite of passive. Passive was going from high to low. This one's going from low to high. And it needs energy. This one requires your cells to use energy. It kind of makes sense, right? Active, you're moving around, you need energy. Passive, you really don't need energy for that. So active transport goes from low to high, and it's the movement of substances across the membrane. But it needs energy. Needs energy, okay? These are going to be large particles. Okay, large particles. The ones where your cell is actually going to have to open its membrane, take it in, envelope it or make it into like a little baggie and transport it around. All right, so Mrs. Todd's going to pass you out a cell, and I need you to cut around it and glue it right here. And I'm going to do the same thing, so I'll be with you in just a few minutes. All right, everybody. If you still need a few minutes, have Miss Todd pause the video and then get everything in your body set up again. And I hope you're going to be able to see why this is active transport and not passive. So I'm going to go ahead and glue my cell down, and we're going to kind of talk about it. Then we have just a few definitions. And I'm off my page, so if you need it to get a new page, go ahead and get a new page to glue this on, okay? Okay, here we go. Now, I want you to take a look down here, down at the very bottom, okay? And I want you to go ahead and circle this word right here. It is endocytosis, okay? Endocytosis. We'll write the definitions of that in just a minute. Endocytosis is taking in of substances. It is taking in of substances. And do you notice what happens? Look what happens. It turns it into a vacuole. And sometimes you can hear it called a vesicle. So either one works. Okay? So endocytosis, think of it as the end of that particle because it's going to get used up. Endocytosis is that cell opening up, letting that substance in, closing around it, and forming a vacuole. Okay? Now, that vacuole moves it around to where that part of that cell needs it. Okay? And as soon as it gets used up, little lysosomes are going to come in and eat it all up and take care of it all. All right? It's going to digest what's left, and then eventually that cell's going to say, hey, you got to go. And it's going to take it and move it, and it's going to let it go. And that out is called exocytosis. Exo, think of exit. Exo is leaving the cell. And I like to think of it as by. You are exiting the cell. Okay? Guys, this is the difference between active and transport, or active and passive. Passive did not require any energy. It was just moving things from low to high. This is active. Your cell actually has to work in order to get this to go. Your cell has to open it up, has to make an envelope or make a vesicle or vacuole around it. Your lysosomes are involved. Your parts of your cell are taking in water, taking in substances, and then after it's used up, it actually leaves the cell again. Okay? So I kind of want you to think of it like when you're eating. You're eating what you need, it gets used up, and for lack of better words, ends up on the other end. Okay? Endocytosis, it's coming in. Exocytosis, it's leaving the cell. Your cell needs energy to be able to do this. And because it needs energy, it is called active transport. Okay? Are there any questions about what we did today? So I'm literally going to take 30 seconds and go through everything real quick, and then I'll go over your lab. All right, here we go. Permeable. Lets all substances go through a membrane. Okay? Semi-permeable only lets certain things go through a membrane. Passive transport does not require any energy for a substance to move from one location to the other location. You're going from high to low. Okay? Diffusion. High concentration to low concentration. May or may not have a membrane, but it doesn't really matter because what drives this is going from a high concentration of a substance to a lower concentration of a substance. No energy is found. No energy is required for this to happen. Passive transport number 2, osmosis. Same type of diffusion. It is a type of diffusion except it uses water through a semi-permeable membrane. So you need a membrane for this, semi-permeable membrane, but you're looking at water for osmosis. No energy required because you're still moving from high to low to find that middle ground of that cell. Active transport needs energy to move substances across the membrane. This is going from low concentration to a higher concentration in the cell. Endocytosis takes in, forms a vacuole or a vesicle, gets used up, gets digested, and then eventually gets kicked out by the cell and eliminated. Exocytosis is leaving the cell. Endocytosis is entering the cell. This requires energy. Active transport. Okay. Again, we'll go over this next class, but we also have a lot of other stuff to cover next class. So read over your notes. Your lab today, let me grab that for you real quick. Mrs. Todd, go ahead and pass the lab out, please. Your lab today is actually going to be witnessing diffusion. On the side cupboards, you're going to notice that there are beakers, that there are baggies, you have cornstarch. I think that's all you need. You're going to need your phones. If you don't have your phones, you can use the iPad or just use the clock on the wall to measure 15 minutes because you're going to need to make some observations after 15 minutes. While you're waiting for those observations, though, you can fill in some of the questions that are here. Okay? And then I need to give you guys a quick definition on this too. So, introduction. In this lab, you will observe diffusion of a substance across a semi-permeable membrane. Iodine is an indicator for starch. An indicator is a substance that changes colors in the presence of a substance when it's indicated. Think of when I blew in the straw the other day. It indicated that there was carbonic acid in my carbon dioxide when I was breathing out. It changed colors. It was an indicator. Okay? Now, here we go. I need you to fill a baggie with a teaspoon of cornstarch and a 100 milliliter baggie or 100 milliliters of water in a beaker. Okay, fill the beaker with 100 mils of water. Add 10 drops of iodine. Place the baggie with your cornstarch in the beaker so that the cornstarch mixture is submerged in the iodine mixture. Wait 15 minutes and then record your observations. While you're waiting, answer the questions. So I want you to go through what is diffusion, what is osmosis, what is the main difference between them, and kind of talk about that kind of stuff, okay? So go ahead and answer those while you're waiting for your 15 minutes. Now, there's a couple questions, there's a vocabulary word I need you to think of. And I want you to circle the word hypertonic. Hypertonic is just a real big fancy word for saying that this is high concentration. So when I want you to think about that, every time you see the word hypertonic, I want you to think of high concentration. There's also hypotonic. Hypo then would mean low concentration. But for now, I want you to think of that. Okay? Answer the questions. They're not easy questions. So you can work with a partner. You have your notes. I want you to really think about this stuff. Okay? Make some predictions. What do you think is going to happen? On the back side, again, make some predictions. Solution in the beaker. What time is it when it starts to turn colors? Make some observations. And then the color after 15 minutes. And then the solution in the bag. What time it starts to turn color. And after 15 minutes, what color is it? And then go ahead and answer the question, okay? This is not an easy lab. You guys are more than capable of figuring it out and answering the questions. Work together. Your lab is due next class. Okay, so basically your lab is you're getting a beaker, 100 mils of water, okay? You're getting a baggie, you're putting, how much cornstarch? Just a teaspoon of cornstarch. You're going to put, and then you're going to seal up the baggie, right? You're going to put that baggie into the iodine. You're going to put 10 drops of iodine in the water, and then you're going to wait and watch and see what happens. Okay? This is not as hard as what you think. Don't get stuck on the vocabulary. We'll go over everything. You guys should have pretty close to half an hour to do this lab, so I would highly suggest you get started. Okay? Sorry again, I can't be there, and I will see you next class.